Well, here we are again, Unit 6, and this is genetics, and we're going to be talking about Gregor Mendel and Mendelian genetics mostly, but we'll make some comments on some other aspects. Gregor Mendel, as I mentioned in Unit 5, studied at the monastery. He was a scholar. He actually was a doctor of philosophy, and he did experiments on thousands of pea plants to determine their traits and how they pass them on from one generation to the next. One of the things that Mendel discovered is called Mendel's Second Law. It's the law of independent assortment that we also talked about when we were talking about meiosis. This is where Mendel got lucky because he concluded that for different pairs of traits, the inheritance of one trait does not influence the inheritance of another trait. So if a bean plant was tall, that didn't have any effect on a bean plant that was hairy or the color of its flower. Well, down here it says, Mendel's second law is genes located on different chromosomes are inherited independently of one another. That's right, that's what we saw, but remember, he didn't know about chromosomes back then. So that's not what he said. He simply said traits are inherited independently of one another. We now know why, and we also know that in Mendel's case, he was lucky because he didn't have any traits that were linked, which means they would travel together on the same chromosome. Every one of the traits he reported on existed on different chromosomes, and that's why he was able to come up with this idea, which basically had to do with independent assortment during meiosis 1. Okay, so if you remember now, in anaphase 1, the homologous pairs of chromosomes line up along the central plate of the cells that are going to divide into two cells, and the one that's from mother or father can assort independently of each other, so that you see here both the mothers are on opposite sides of this and the fathers are on opposite sides of this, but it could be the other way around. Both the purple ones could be on top, both the green ones could be on bottom, or vice versa. And that's what we mean by independent assortment. So, you know, you could be 90%, have 90% of the genes came from your father that you have, or 90% of your genes could be from your mother, or 60%, 40%, or 25%, 75%. It's all happens at random, and when these things assort independently on different sides of the central plate, and they'll be pulled to different parts of this cell to become two cells, so that these two will be totally different. Okay, so there is a video that shows you how to do this lab for Unit 6, but I just wanted to remind you that what we're going to do is we're going to have flies that we're going to cross. We're going to segregate their alleles. Parent 1 will be, in this case, let's say LL, a homozygous long-winged fly. Well, there's homozygous, both the same. All sperm the same, okay? And then parent 2, we'll call her the female, a heterozygous long-winged fly. Okay, LI, so small l and big L, so heterozygous. So one egg is dominant long-winged and one is short-winged, the small l. Check parents, yep. So then this is how we do a Punnett square, and then we start mixing them up or putting them together here. So this one is going to be LL, right? This one is going to be LL, same thing. This one is going to be LI, right? And this one's going to be LI. So we're going to get a 50-50 mix according to genotype. But phenotypically, look, they're all going to be the same because of dominance. And then the other part of this lab has you looking at gray bodies versus black bodies, and gray is capital G, and black is small g, okay? So we're looking at traits that are dominant or recessive, and if they're recessive, they have to be double recessive to be expressed. Otherwise, if there's one dominant allele in your makeup, that's what you will be phenotypically. In other words, you will express that, you will look like that, you will act like that, you will have that trait, whatever it may be. Okay? So when we go to the lab journal, there are some questions. And of course, you're going to submit those just like you've been doing. But let's go to the fourth question. I want to talk about that a minute. The fourth question, what genetic information can be obtained from a Punnett square? What genetic information cannot be determined from a Punnett square? Well, everything we've done with Punnett squares has dealt with discrete traits that are independent of each other, and that by discrete I mean it's either tall or it's short, it's either pink or it's white, or whatever. One or the other. One's dominant, one's recessive in terms of the alleles. That's the kind of stuff we use Punnett squares for. We can use it to look at one trait at a time, or we can use it to look at multiple traits as long as one is dominant and one is recessive. But there are other kinds of traits that we have genetically. Mendel didn't look at them. We learned about them later. And if you read the second part of this question, you read page 166 of your book, you'll run into the answers to this question. You'll also run into it on the PowerPoint. So let's take a look. Okay, so some traits are continuous. For example, how tall people are. You don't have tall people and short people. You got people that are short, and then you've got them taller and taller and taller and taller, all the way to the maximum of 8 foot 6 inches. I think that's approximately what the tallest man in the world is, and he lives in Russia, and there's another one in Korea that is over 8 foot also. Anyway, those are continuous variables, and they're represented. We can represent them by looking at a bell graph like this, or a diagram. And here we have people being set up in this size category or height category just to demonstrate what we're talking about. And then we have the graphic below that shows that it's sort of a bell-shaped distribution. This is a continuous variable, and this is one that, you know, you can't do with a Punnett square. Then there are pleiotrophic effects. Your book talks about that. It's when an allele or a gene has more than one effect on a phenotype. You know, there are a lot of disorders like sickle cell disease, cystic fibrosis, and so forth that are examples of this. Are there other examples? Sure there are. Codominance. A gene might have more than 2 alleles in a population. So, what kind of examples would we have of that, do you think? Well, we're going to talk in a minute about blood groups. But let's cover one more thing before we do. Let's talk about incomplete dominance. Okay? Incomplete dominance is where, for example, we have traits that merge, sort of. So we have pink and white flowers, in this case, that become purple, or somewhere in between pink or red and white. Another example is human beings. When a black person marries a white person, we get all kinds of shades of black or brown, everything in between white and black, because those are incomplete dominance genes. And you can't do Punnett squares with them because you don't know if somebody's black and somebody else is white, what you're actually mixing together and what the result will be. You just have to wait and see. Surprise, surprise. So let's go back to the dominant recessive genes, but let's go talk about the ones that are co-dominant, okay? Co-dominance is when different genes have more than 2 alleles in a population. So, and these alleles are equally dominant. So if you have both of these alleles, you have both of these traits. The primary example that should be of interest to you is blood groups. You can be AB, you can be A, or you can be O. A, B, AB, or O. You can also have an Rh factor that is positive, which means you have it, or you don't have it, and we call that Rh negative. So let's make another video and let's talk about blood groups. But before we do, let's take a look at one other thing, some background information I want you to have. Okay, there's some further background information that I've posted links to. There's one on the left-handed gene, which is an effect that does not follow the dominant, co-dominant, discrete variables, because if you are left-handed, what it really means is you are not right-handed. You watch that video and you think about it. It's a short video. And then there's about a 12-minute video, which is Mr. Anderson's video on a beginner's guide to Punnett squares. And that's a pretty good one because it teaches you more about how to use Punnett squares for discrete variables. It's worthwhile watching. These are posted under there's a link, and this Word document is attached to the Unit 6 lecture tab. Then finally, if you want to watch a PBS NOVA video that's on something really cool, it's on epigenetics. And what's really cool about epigenetics is it talks about the genes that are formally called silent genes, which actually regulate genes. Turns out we have the same gene for our nose as an elephant does, but the silent genes regulate whether our nose turns into a nose, or whether it turns into Pinocchio's nose, or whether it turns into an elephant's trunk, okay? And epigenetics also talks about the differences between identical twins that cause them to be actually not identical at all, because as they grow and develop, their genes change in response to environmental factors like things that cause mutations, or other factors like smoking, diet, and chance alone that makes their genetic makeups diverge over time as they get older. So only at the very beginning of conception are identical twins identical. After that, they're not identical anymore, actually. That's a pretty fascinating video, and it's worthwhile watching. It's an excellent video.